Hello, everyone, and welcome to today's broadcast, Integrative Cancer Immunology and Novel Concepts of Cancer Evolution, presented by Dr. Jérôme Galon, Research Director, French Institute of Health and Medical Research. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speakers will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Galon. I will turn the presentation over to him.
Thank you. Hello, everybody. So I'm going to discuss today a novel concept of cancer evolution and the importance of immunology in cancer. So here are my disclosures. I'm the co-founder of an immuno-oncology diagnostic company, and I do talk to people in industry. So as you all know, the definition of cancer is based, and the severity of cancer is based on a traditional staging system, basically showing that the cancer is progressing into a primary organ, and that is the T stage. And additional parameters like the invasion into the node or the distant metastases are essential key elements to classify cancer patients into early stage disease or late stage disease. We all know cancer is a genetic disease due to mutation, to alteration of the genome, to chromosomal instability, microsatellite instability, etc. And these tumor aggressiveness parameter, the progression of a cancer, the invasion, defines early and late stage cancer. And of course, patients are treated accordingly to their stage. What I'd like to discuss today is a totally different concept and a novel paradigm for cancer, where in fact we propose that there is a dynamic interaction between tumor cells and immune cells. And in fact, what we showed was that all these key criteria of tumor progression, the invasion of a cancer, the metastasis, the recurrence of a cancer, are all dependent on the pre-existing immunity and on Immunoscore, that is the measure of T cells inside a tumor. In fact, the fate of the tumor cells is dependent on the pre-existing immunity and on T cells. The pre-existing immunity of the patient is determining the fate and the survival of the patients. And now that we have efficient immunotherapies, what we see is that this pre-existing immunity and the T cell response is also determining the likelihood of response to immunotherapy. So I will not focus, or at least at the beginning, on a particular molecule, on a particular pathway. I don't have any favorite cells. But really what we wanted to do was to decipher and to analyze deeply in a comprehensive manner the tumor microenvironment in large cohorts of human patients, to try with systems biology to understand what is the tumor microenvironment and how this is important. So here is an example of a tumor, a cancer, colorectal cancer. You can see in the middle the tumor cells, on top the normal epithelium. Surrounding this tumor, you can see massive tertiary lymphoid structures. And inside the tumor and surrounding the tumor at the invasive margin of the tumor, you can find basically every single subset of immune cells, all subsets of T cells, T helper cells, Th1, Th2, Th17, regulatory T cells, all subsets of memory T cells, of B cells, all the subsets of innate cells. They are all present in all cancers at different densities, different levels, different locations, different activation status, but they are all there. So the first thing we asked was, really, what is the importance of this pre-existing immunity in human tumors? Does it really matter? And what we found back in 2005 in this article that we published in the New England Journal of Medicine was that indeed there are important immune parameters that are associated with the presence or absence of early metastatic invasion signs, that is the presence of tumor cells inside the blood vessels or the lymphatic vessels. And in fact, what we found was that there is a good immunity, in particular memory T cells, and in particular effector memory T cells, that correlated with the absence of this early metastatic invasion, preventing those, and correlating with an improved clinical outcome in the patients. Here is an example of some of the studies we did. We did large FACS phenotyping, looking at fresh tumors. On the left side of the slide, you can see in each column here represent one tumor from one patient, and you can see more than 400 subpopulations of immune cells analyzed by flow cytometry from those fresh tumors. And when we looked at the patients having or not having these early metastatic signs, we could see that there are indeed some T cell subpopulations that are significantly different between the VLP positive patients and VLP negative patients. In particular, markers of T cell activation, T cell differentiation, the Th1 subsets, and the memory T cells. So we went deeper into the analysis of these memory T cells and these cytotoxic T cells and really quantify them in a deep manner. And this led us to publish this paper in Science, which was the foundation for this new concept of immune-contexture that I will describe, and for this assay Immunoscore. So what we did at the time was the quantification with digital pathology of more than 6,000 immunohistochemistry, quantifying the densities of different subtypes of adaptive immune cells within the tumors, taking into account not only the type of immune cells, not only the density, but also their location in different areas of the tumor. So here is the survival curves of the patients according to the international AGCC, UICC, TNM classification, based on tumor histopathological findings. This is the current gold standard classification. You can see on the x-axis here more than 15 years of follow-up, and you can see patients relapsing. Stage 1 patients having small tumors, T1, T2, stage 2 patients having more advanced T3, T4 tumors, patients with stage 3 having lymph node metastases. Now, when we did the quantification of the T cells, and in particular the memory T cells, taking into account where they are located in the center, here CT, or in the invasive margin, here IM, we quantify the densities of memory T cells in the invasive margin and the densities in the center of the tumor. What we found was that the patients with a high density of memory T cells in both tumor regions had an excellent outcome. As you can see, more than 80% of the patients had no tumor recurrence for more than 15 years. And this was happening regardless of the stage of the patients. Even a patient at a late-stage tumor, what is considered today a late-stage tumor, those patients were protected and did not get tumor recurrence. In the opposite, the patients who had low densities of memory T cells in both tumor regions had a dramatic outcome, as you can see, regardless of their tumor stage. Even patients at an early stage would have a very rapid tumor recurrence, which means that the coordinated adaptive immune reaction, measuring the T cells inside the tumor, more than the classical tumor invasion parameters, really predicts the clinical outcome of the patient. So this was showing for the first time that the pre-existing immunity and the T cells, when put in a Cox multivariate analysis, and this was shown for the first time for any cancer, that this evaluation of the immune cells, which is in fact the Immunoscore assay, is highly, highly significant in a Cox multivariate analysis. This multivariate analysis is testing the strength of the parameters and the dependency of the parameters. And not only Immunoscore is highly significant, but for the first time, what we found was that the T stage, the N stage, the grade of differentiation of the tumor are no longer significant. They are all statistically dependent on the Immunoscore, on the T cell pre-existing immunity that the patients have in their tumors, which brings a novel paradigm for cancer, because now this classical classification of cancer is no longer significant. So we decided to put this into the concept of immune-contexture. So the contexture is the act of assembling parts into a whole, or the arrangements of interconnected parts. And so these four important parameters in terms of the immune microenvironment are the nature of immune cells, the type of immune cells, the immune functional orientation of the response, in particular Th1 cells, the density of the immune cells within the tumor, and the location of these immune cells, in particular at the invasive margin of the tumor and in the tumor center. And we propose that these are essential parameters, which we analyze more deeply into this paper that we published back in the GCO in 2011. On this paper, we analyzed the reason why, on a Cox multivariate analysis, when we put all the key criteria, the key pathological findings, the T stage, the N stage, the number of lymph nodes, the histological grade, etc., all those parameters are no longer significant and are statistically dependent on the pre-existing immunity, on the pre-existing T cells measured by Immunoscore. Only bowel perforation, as you can see, remains significant, which is quite bad, and Immunoscore. And a direct comparison of the AJCC-YCC TNM classification with the Immunoscore classification for DFS, overall survival, disease-specific survival, shows the power and the significance of the Immunoscore classification and the dependency of the TNM classification on this Immunoscore classification. And together with our paper, we had this quite provocative editorial, TNM staging. T is for T cell and M is for memory. So back in 2006, when we published this paper, we put patients into major categories of tumors in terms of immune infiltration. The high Immunoscore, the optimal immune response, also called inflamed tumors or hot tumors, are the ones having long-term survival. The absence, the low Immunoscore patients, also called non-inflamed or cold tumors, we put them into these big two categories. We also did three categories with this altered immune reaction, this intermediate Immunoscore, having an intermediate immune response, which we also subdivided into two mechanistic distinct alterations. Either the exclusion phenotype, which is due to the accumulation of T cells at the invasive margin of the tumor only, versus the altered phenotype that is due to an immunosuppressive mechanism. And this is actually the figure of the paper that we published back in 2009, way before immunotherapy was so popular, showing that, in fact, the risk of relapse at 2 years, when the patients have an optimal immune reaction on top with a high Immunoscore, hot tumors, these patients have less than 10% risk of relapse at 2 years. When they have an altered immune response, the risk of relapse at 2 years for those patients is 50%. It can be due to two distinct mechanisms, either this immunosuppressive phenotype or this exclusion phenotype with a loss of coordination and T cells only surrounding the tumors, not getting in. The worst case scenario, the patients having low Immunoscore, no sign of T cell response, these cold tumors, they have a risk of relapse at 2 years of 80%. And we published that back for the first time, this categorization of patients based on immune parameters in 2009. We are now more and more refining this immune-contexture, so consisting in these 4 categories: the type of immune cells, the density, the location, and the functional immune orientation, basically measuring the cells, quantifying them, taking into account the spatial localization, but also measuring the quality of the immune response with immune signatures. And you can see some of the important immune families here. We also did a study to evaluate whether the evaluation of immune cells, in particular cytotoxic and memory T cell densities, could also predict early stage risk. And it is known for colon cancer that patients with stage 2, stage 1, 2 would have about 20% risk of relapse, which we can see on these survival curves here. This dotted line shows that there is about 20% of the patients who will experience a relapse, even though they have a small tumor, stage 1, 2. And this is 18 years of follow-up. Of course, these patients, these 20% of patients, would be important to know which one they are, because there is no recommendation for adjuvant treatment. These patients have their surgery and most of them go home. So evaluating the Immunoscore, that is the quantification of the cytotoxic T cells in the center and invasive margin of the tumor, showed the following results. Patients with an Immunoscore 4, so high densities of memory cytotoxic T cells in both regions, high, 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 those patients have an excellent outcome. More than 95% of the patients will survive with no recurrence for 18 years. They are safe. But the patients with Immunoscore 1, 2, or even worse, the yellow curve, Immunoscore 0, these patients have very high risk of tumor recurrence, even though they have an early stage tumor. So it is possible to see, based on the T cell evaluation and the T cell density with this measurement, what are the high-risk patients. And again, when we do the Cox multivariate analysis, what we can see is that the T stage is dependent upon this immune evaluation, Immunoscore. So of course, this could have very important clinical implication, and we know now that we have immunotherapies that immunotherapies are working and are not working on all patients. And when you have these 2 types of patients, the cold tumors, Immunoscore 0 with no T cells, these patients are very unlikely to respond to a checkpoint inhibitor, anti-CTLA-4, anti-PD-1, that are targeting the T cells. These patients with cold tumors, they first need a T cell priming before to come with a checkpoint blockade. In contrast, the patients with Immunoscore 4 tumors, full of T cells, pre-existing T cells, are the most likely to respond to anti-CTLA, anti-PD-1 treatment. But biology is not as simple as these 2 extremes. And in fact, with the Immunoscore evaluation, we can measure all the subtypes and densities of immune cells, of immune T cells, from Immunoscore 0 to Immunoscore 4. And we can also highlight the different immune defects in different patients. So we developed software, one of which is Clugo, that is what we published in Bioinformatics. It's the #1 most downloaded software on the UCSD website, with more than 100,000 downloads, basically facilitating the biological interpretation of large data sets. And we also created another software, Clupedia, that is also published in Bioinformatics, also a lot of downloads, basically adding to your experimental data the knowledge of multiple databases that are publicly available in order to add data and knowledge to your experiments. So we did that using our tools, Clugo and Clupedia. And what we did was to ask the question, what makes the difference between patients having a hot tumor, Immunoscore high, versus a cold tumor, Immunoscore 0? And here is a list of genes that we found to be significantly associated with high and low Immunoscore and with the survival of the patients. So each node represents one gene. The color of the gene represents its function and the connections, the correlation between those genes. And to these data, we added conserve genomic neighborhood data from publicly available databases, phylogenetic profiling from public databases, co-expression analysis, protein-protein interaction, receptor-ligand interaction, functional genomics databases like Affymetrix data, TCGA data, etc., to do in silico prediction. And we end up showing that a network with some bioinformatics prediction. As you can see from those predictions were two families of molecules, the chemokines, including CXY-CL1, CXCL9, CXCL10, CXCL5, and adhesion molecules. And so with these hypotheses, now we went back to our patients and did the experimental validation to see whether those predictions and chemokines and adhesion molecules were important, and reported in this paper back in 2010 that indeed these are essential molecules to attract and to get increased densities of T cells inside the tumor. So we highlighted several mechanisms that are associated with T cell infiltration, the attraction of subtypes of immune cells with different subtypes of T cells that are attracted by different chemokines. And in another paper published in Science Translational Medicine in 2014, we highlighted another mechanism explaining the high densities of T cells through the local lymphocytic proliferation that is mediated by the level of expression of a cytokine IL-15 inside the tumor that is basically expanding the pool of the activated T cells, basically giving two immune-related mechanisms that are associated with T cell infiltration, high Immunoscore, and hot tumors. We also went into the analysis of tumors to see what is the spatial temporal dynamics of the evolution of the immune response with the tumor progression, because this is not a static endpoint. The tumors are progressing from T inside 2 to T1 to T2 to T3 to T4. In parallel, there is an evolution of the immune response. Different immune cell types are present, different activation level and immunosuppressive mechanisms are in place, and we wanted to evaluate this spatial temporal dynamics. And we end up showing the immune landscape in human tumors in this paper published in Immunity in 2013. In fact, what we did for the first time was to evaluate the immunome, that is the specific immune gene signatures coming from purified immune cell subpopulation. So in each column here, you have different immune cell types. For example, at the beginning here, top left corner, B cells, and then T cells, and then subsets of T cells. And you can see, for example, neutrophils, mast cells, macrophages, etc. And you have here the main genes and the most specific genes that are upregulated only in one specific immune cell type. And we applied this immunome, this is something now that is very popular, to the human tumors to basically evaluate the immune composition of a tumor coming from a complex mixture of cells by deconvolution of the complex expression profiles. And this is very popular, of course. These have advantages and disadvantages, and we validated our findings by doing the real quantification of all those different immune cell types by immunohistochemistry and quantification by counting the number of cells per square millimeter of tissue, basically measuring the immune cell density. So here, each peak represents the height of the peak represent the cell density, immune cell density. You can see a network representing the correlation and the same behavior between the cells. And you can see two subnetworks, the blue one, that is the quantification in the center of the tumor, versus the yellow one, that is the quantification at the invasive margin. You can also see subnetworks here, and you can see that the density is very much dependent upon where you are quantifying those cells. For example, B cells are found at very high densities on the right side here at the invasive margin. And on the far left, you can see the B cell quantified in the center of the tumor, very low density. You can also see cells that are outside of the network, like the granulocytes, the R17 producing cells, and having similar behavior. Now, instead of plotting the density, which is very informative, we looked at the evolution of those densities with the progression from T1, T stage to T2, to T3, and to T4 stage, and could show that indeed there are some immune densities, immune cells that are coming into the tumor, some that are decreasing when the tumor is progressing. And then we look at the impact of each of those immune cells on the survival of the patient. Instead of plotting the hazard rate, the densities, like here, now we are plotting in the same network the hazard ratio, that is the impact on the survival, the impact for each of those cells. The green peaks represent the good immune cells, high densities correlating with prolonged survival. And you can see that these major network are all green peaks except the two little bad peaks of CD68 macrophages that are slightly bad here. But all the subsets of T cells, except the R17 producing T cells, all the subsets of T-follicular-helper cells and the B cells are associated with a prolonged survival. And we could show the difference between T1, T2, T3, T4 of those densities and show how each of those cells were changing from one state to the other. Basically showing that the adaptive immune cells, including the T cells, CD8, CD3, CD57 activated T cells, memory T cells, cytotoxic T cells, FOXP3 T cells, were decreasing with the progression of the tumor. In contrast, there was an early increase in densities of many innate cells and T-follicular-helper cells, and a late increase in the B cells. The cells, immune cells that were associated with tumor recurrence are the T cell subsets, including the Th1, the cytotoxic, and the memory T cells, the T-follicular-helper cells, and the B cells, all being good immune cells. And so we put this idea that there are important immune parameters coming from these tertiary lymphoid structures surrounding the tumors, acting through the T-follicular-helper cells that are producing the cytokine IL-21 and the chemokine CXCL13 that are important to shape the proper immune-contexture inside the tumor, in particular, the generation of the good cytotoxic and Th1 response. So going deeper into the analysis of the tumors, human tumors, we were very interested by a subgroup of patients that have a specific type of genetic alteration. These patients have a DNA mismatch repair deficiency. They're also called MSI patients for microsatellite instability. The characteristic of these patients, because of their DNA mismatch repair deficiency, is that they are unable or cannot repair mutations that are appearing on the genome, in particular frameshift mutations. And frameshift mutations are very interesting from an immunological point of view for two reasons. One, because they are not random. The frameshift mutations happens on the genome on repetitive sequence on the genome. Like a suite of A, very often there is an additional A. And when the DNA mismatch repair is deficient, it cannot be repaired. And for an immunologist, this frameshift, this addition of the new A, shift the code, and therefore the protein becomes completely different. And this new protein generates new peptides that are totally different from self-peptides. And therefore, many of those are immunogenic and can be detected by the immune system. So we analyzed the mechanistic relationship between the tumor genotype, the microsatellite instability status of the patients, and the immune response, and in particular, Immunoscore. We quantified two large cohorts, including one cohort of more than 200 patients from the TCGA and a cohort of more than 600 patients from our hospital. We did multiple different types of approach, basically to analyze deeply the mechanistic relationship between the microsatellite instability status of the patient and the generation of an immune response. What we found was that looking at the genomics, on top you have in blue the MSI patients and in white the MSS patients. Looking at the chromosomal instability, here on top is blue and red lines, and then looking at the mutation profiles of those tumors with all the mutations, main driver mutations in that case here, and looking at the transcriptome of those tumors, looking at all the genes that are significantly different between MSI patients and MSS patients in the tumors. We found that there are more than 400 genes that are increased in MSI tumors and more than 400 genes that are decreased in MSI tumors. When we look at the function of all those genes across the genome, we were surprised to see that there were no major green functions, meaning downregulation of genes who are not belonging to a particular function. But for the upregulated genes, as you can see from all those red nodes, they belong to consistent families of molecules that, as you can see, are very relevant for the immune system. All those genes, or many of those genes, the majority of those genes were immune-related genes from the interferon gamma mediating signaling pathway, from the antigen processing and presentation, from peptides, antigen positive, regulation of leukocyte, migration of immune cells, response to TNF, etc. So we looked whether those mutations were appearing, and indeed, as you can see from this log scale number of FS frameshift mutations, you can see that patients with MSI do have more frameshift mutations than patients with MSS. And as I told you, these mutations are not random. And so we predicted the top 20 regions on the genome having repetitive sequence where there is a very high likelihood of having a frameshift mutation. And we validated these in an assay here. And as you can see in blue now is the validation of the frameshift mutation. Indeed, we found frameshift mutations here at the bottom of STA1 gene, of TAF1B gene, TGF-beta-R2, very frequently having frameshift mutations, in contrast to MSS patients not having those mutations. The beauty of that is that we could, with this, compare the observed versus the expected number of frameshift and missense mutations, and also the observed and expected number of immunogenic mutations. By doing exome sequencing, RNA sequencing, the detection mutation, the variant calling, finding the HLA haplotype of the patients, doing epitope binding prediction, HLA TCR binding prediction, giving basically an immunogenicity score for each of the mutations. And we could compare the silent mutations, those that are not changing the amino acid and are irrelevant for the immune system, versus the non-immunogenic mutation versus the one with a high immunogenicity score, these immunogenic mutations. And so we compare the rate of mutations and silent mutations across all the genes from all the patients from the TCGA database, pan-cancer, so more than 3,000 patients tested for the number of silent mutations. And we compared it to the rate of non-immunogenic mutation versus the number of expected immunogenic mutations versus what we finally observed when we did the sequencing, and should show that indeed patients with MSI have increased or have significantly less immunogenic mutations than what is expected based on the rate of silent and non-immunogenic mutations. Similarly, missense mutation was found to be significantly less in MSS patients and in MSI patients, basically showing with genetic evidence that immunoediting is occurring in human tumors, that there are less than expected immunogenic mutations based on genetic sequencing. So if this is the case, then we should be able to detect T cells that are specific for frameshift mutations. So what we did was to take the T cell from the patients, to sequence those patients, detect their HLA haplotype, restricted to HLA-A202, and looked at those having in that case, a TGF-beta R2 frameshift mutations. And we did that for some other frameshift mutations. And what we could see is that for MSI patients, the T cells from these patients are able to kill on this chromium 51 assay to kill tumor cells presenting the frameshift mutations, whereas the T cells from an MSS patient or the T cells from a patient that don't have that mutation are not able to kill those tumor cells and those tumor cells having the mutated peptides. Therefore, it is possible to detect anti-tumor specific T cells in those patients and anti-tumor specific frameshift peptides in those patients. So there is a mechanistic relationship between the increased frameshift and missense mutations found in MSI patients and the likelihood of generating an anti-tumor specific cytotoxic T cell response. But when looking at the survival of the patient, what really mattered at the end of the day for both overall survival on the left side or disease-specific survival on the right side here is the fact that the patients have generated a strong adaptive immunity measured by Immunoscore. So here, Immunoscore 3 and 4, the top two curves, blue and pink, are the patients with a high Immunoscore, whether they are MSS or MSI. Vice versa, the patients with a low Immunoscore, Immunoscore 0, 1, 2 here, yellow and white curves, shows bad overall survival and bad specific survival, whether they are MSS or MSI. And the multivariate analysis shows the dependency of MSI on Immunoscore, Immunoscore being highly significant on a multivariate model. So the only difference between MSS and MSI here is the frequency. More frequently, patients with an MSI can generate a strong adaptive immunity, and more frequently, they will have a high Immunoscore. But when they don't, then those patients are not doing well. So the fact that T cells are essential is likely to be valid for multiple cancer types because the immunity doesn't care where the tumor cells are located, and immune cells are infiltrating all sorts of tumors. And indeed, when doing a meta-analysis of the literature looking at the prognostic value of subtypes of T cells, what we found that was published in that review in Nature Review Cancer was that the tumors that are infiltrated with high densities of cytotoxic CD8 T cells or memory CD45RO positive T cells showed with this blue histogram the good prognostic value of these cells. In most of the study, here you have the percentage of articles published or the number of published articles. 58 out of 60 articles showed the significant good prognostic value of cytotoxic T cells. Similarly, for Th1 being associated with a good outcome. But other subtypes of T helper like Th2, Th17, or regulatory T cells could be either good or bad or with no significant impact depending on the cancer type, and it was studied for more than 20 different cancer types. So is this quantification of the pre-existing immunity of the T cells with a standardized assay like Immunoscore clinically relevant? We believe so because when looking at the median overall survival of the patients, for patients having a high Immunoscore, Immunoscore 4, with the measurement of the cytotoxic T cells in the center and invasive margin, those patients will survive more than 15 years. regardless of any other parameters. Immunoscore 2 patients will survive 5 years, Immunoscore 0 will survive less than 2 years. Major clinical differences. But the reality of clinical practice today is that it is not taken into account yet. So we went for an easy and simple, powerful immune test, Immunoscore, and to test whether this could be applied in routine practice. The current situation for all patients with cancer is that only tumor cell characteristics are measured. Of course, the cancer classification based on the TNM staging system, plus multiple ways of classifying tumors and patients based on tumor cell characteristics, either anatomopathology, morphology-based type classification, either based on the tumor cell of origin, either based on molecular pathways like chromosomal instability, microsatellite instability, based on the mutation status of the patients or on tumor gene expression signatures. But there is not a single assay that is done in routine to tell us something about the immune system of a cancer patient. We don't know anything about the immune status of a cancer patient. There is no measurement of the immunity to cancer. So the idea of Immunoscore is to have a first host immune response evaluation quantifying the densities of cytotoxic T cells in the center and embedded margin of the tumors. We propose to do that and wrote this review to propose to include into the classification of cancer. We initiated with the SITC, the Society for Immunotherapy of Cancer, a consortium followed by multiple cancer societies and immunotherapy societies, and initiated this worldwide Immunoscore consortium. These many countries replied and were interested in participating. We did a lot of harmonization to decide what should be the Immunoscore assay. And it's basically the quantification of all those brown T cells that you can see here and to count them one by the software. So basically what we did was that after harmonization of the assay, that all centers performed the Immunoscore on their patients. The data were QA'd and sent to an external statistician outside of the consortium at the Mayo Clinic. All the clinical data were sent to a US site that encrypted the data and sent it to the Mayo Clinic. And the statisticians divided the patients into 3 cohorts. So basically, what we're doing is a standardized quantification of cytotoxic T cells and T cells inside regular whole slide sections where every single T cell here is counted. And for you to know, the average, the mean number of T cells on a given slide, on a given tumor is 88,000 T cells. Huge numbers. And this was validated, and we just published this year the result of this international study for the consensus Immunoscore to classify colorectal cancer patients. Here is an example of the densities of immune cells that were found in more than 3,000 tumors, quantified huge densities of immune cells. You can also see the huge difference, more than 3 logs difference in the densities between a tumor and another tumor from another patient. And all primary endpoints, secondary endpoints, survival data were significant. Here are the results of the multivariate analysis showing that Immunoscore 2, 3, or 5 categories show highly significant p-values in multivariate analysis, both for time to recurrence, disease-free survival, overall survival. And this is the relative contribution to the risk test, showing on the left side the current situation with all the parameters that are evaluated today and their relative contribution to the risk. The end stage and the T stage are the most important. Grade of differentiation, venous emboli, lymphatic invasion, perineal invasion. Now on the right side, you see the same test by adding Immunoscore, showing the power and the relative contribution to the risk of Immunoscore in comparison to all the other parameters. Basically, this international validation of the immune consensus Immunoscore for the classification of colon cancer gives strong arguments for introducing for the first time an I, like immune, into the classification of cancer, TNMI. We have multiple arguments and level of evidence to support this. But really what's is struggling is interesting is really at the metastatic state, because is there an impact of the immune response at metastatic stage? So what we did was to analyze the M-stage patients, or the primary tumors from patients without metastasis or with metastases. And we published that in Science Translational Medicine to see what are the parameters that are associated with the dissemination to distant metastases. We analyzed multiple genetics, chromosomal instability patterns, mutation, tumor profiling, and immune profiling by quantifying the immune microenvironment to ask the question, what in the primary tumor is associated with early metastatic dissemination, synchronous metastasis, or metachronous metastasis? And basically could show that there are no or not much going on in terms of genomic patterns associated with metastases. But in contrast, there's huge differences in the microenvironment that are associated with or without the dissemination to distant metastases. Two of the most striking findings are the densities of the cytotoxic cells, immune cells, granzyme B positive in the center of the tumor, preventing metastasis dissemination, and the densities of lymphatic vessels at the invasive margin of the tumor, preventing metastatic dissemination. So is there a metastatic, at the metastatic stage, an immune escape? So we did two studies to evaluate the immunity within metastases. In this first study, we took only the same type of primary tumors, colon cancer, giving liver metastases or lung metastases, and analyzed more than 400 metastases, and performed the Immunoscore, the consensus Immunoscore, on the metastases, not on the primary tumors. What we found was that high densities of cytotoxic T cells and high Immunoscore correlated with these blue curves with a very prolonged overall survival. As you can see, 5 years, more than 70% of the patients are surviving 5 years. In contrast, low Immunoscore tumors, metastases, those patients have only 20% chance of overall survival at 5 years. We picked the worst case scenario of patients having brain metastases. In that case, the study was any type of primary tumor, the opposite study, giving rise to only one type of metastasis, brain metastasis, and we studied the immune microenvironment within the brain mets and the consensus Immunoscore within the brain mets. You can see T cells surrounding the brain metastasis, you can see T cells inside the brain metastasis, and you can see with this long-term follow-up on those patients having had brain mets that the patients with a high Immunoscore in white here have very prolonged long-term survival, more than 5 years, as you can see here, whereas the patients with a low Immunoscore have much shorter survival, showing that even in the worst-case scenario of patients having brain mets, the Immunoscore within the metastasis predicts survival. We followed this by a study to really analyze the heterogeneity of the mets. And we analyzed lung metastases, liver metastases, peritoneal metastases, more than 600 metastases from 2 cohorts of patients, quantifying multiple densities of immune cells, which we reported. So it's a full quantification of resected metastases, much larger than a biopsy, of course. It's a full metastasis quantification, the whole invasive margin of the whole metastasis and the whole center of the metastasis. And basically what we showed, and this has been accepted for publication in Cancer Cell, is that there is an heterogeneous pattern of metastasis. Patients with different metastases have different immune microenvironments, that the tumor cells are different, and also the immune cells are different. And that in fact, what really matters for the survival of the patients is the density and the strength of the immune response in the least immune infiltrated metastasis, that is the one that will ultimately kill the patients. We also could analyze the difference between biopsy analysis in metastasis versus whole slide analysis and whole metastasis analysis, and could show that in fact the accuracy prediction of different markers, for example PD-L1, on biopsy was really poor in comparison to the Immunoscore evaluation, which was much better in accuracy prediction in comparison to the whole metastasis. Then, lastly, what drives metastasis? What are the mechanistic escape mechanism, and is it compatible with the current theories of cancer evolution? So what are the current theories of cancer evolution? There are four main models. The linear model, stating that there's an accumulation of different driver mutations leading to cancer development. The neutral model, saying that there is no impact of the type of driver mutations and no selection. The Big Bang model, saying that there is an initial major Big Bang of mutations and accumulation of many mutations before the dissemination. And probably the most accepted branch model of different drivers giving rise to different clones with a branch evolution. The common characteristic of all those theories is that none of them involved a role of the immune system and a possible immune pressure through Darwinian selection based on immune parameters. So what we did was to analyze not only primary tumors, but from very special patients having synchronous metastases and long history of metachronous recurrent disease with multiple metachronous recurrence for more than 11 years. We analyzed deeply by doing sequencing, whole genome sequencing and RNA, looking at the clones, and did the tumor clone quantification, the evolution of the tumor clone, the prevalence of those clones within the different metastases, could follow and trace back the clones, basically showing the phylogenetic tree of the clones. Beyond the phylogenetic tree, could show the evolvogram of those tumor clones, so showing that some tumor clones are persistent in the different metastases, but some are not persistent. And what we found was that, in fact, the non-recurrent clones are immuno-edited, whereas the progressing clone are immune privileged. We could also see that here that the clones within the metastasis have a much higher immuno-editing score. Lower score means more immuno-editing, and that those clones were eliminated and not reappearing more than 4 years after. In contrast to the persistent clones, all of them showing no signs of immuno-editing. And at the end of the day, when analyzing all the parameters, the number, the type of driver mutations, etc., what really was associated with metastasis recurrence were the four following parameters. The Immunoscore of the metastasis, so the quantification of the cytotoxic T cells inside that particular metastasis. The immunoediting happening in that metastasis, the distance between the T cells and the ongoing proliferating Ki-67 positive tumor cells, and the size of the metastasis. And these four parameters are the ones remaining significant on the multivariate model, explaining the future recurrence of the patients and of the metastasis and of the clone who will have a metastasis. So this evolvogram showed that there is a multiverse of metastasis evolution in space and time under immune selection pressure, that there is an evolution of tumor clones that is linked to the intrametastatic immune-contexture, and that the non-recurrent clones are immuno-edited, like these ones, unlike the progressing clones that are immune privileged. And we propose that there is a parallel selection model which describes this clonal immunoediting, and we propose a parallel immune selection model to describe tumor evolution during the metastatic process. So immunoediting and Immunoscore are the predictive factor of metastasis recurrence, as well as the distance between T cells and proliferating tumor cells. And we propose a novel theory of cancer evolution, a parallel immune selection model, which basically takes into account this immune pressure in each of the tumor microenvironment that is acting on the selection of tumor cells. And we propose that there is a dynamic interaction of tumor cells with immune cells and a Darwinian selection of immune escape, and we could show this with a parallel evolution and multiverse of metastases. So as a conclusion, nowadays, immunotherapies is becoming the fourth pillar of cancer treatment since the approval, the recent approval of ipilimumab, the anti-CTLA-4 T-cell blocker acting on T cells, and since the approval, the breakthrough of the year in 2013 with anti-PD-1, another checkpoint acting on T cells. Now there is an expansion and an approval month after month of different checkpoints targeting T cells on different cancer types. And these checkpoints targeting T cells are releasing the brake, are breaking tolerance, are reactivating the T cells. These T cells I just talked to you about, this pre-existing immunity that we showed to be so important. And we are able to unleash this pre-existing immunity with agents that are targeting these T cells. Then we have this clinical success, and also the clinical success that was announced yesterday of the Nobel Prize for Jim Allison and Tasuku Honjo, who worked on the CTLA-4 and anti-PD-1, showing that it is possible to release the brake from the T cells and to unleash the pre-existing immunity with the manipulation of those T cells. We expect that this will be happening in multiple different cancer types. And hopefully, these are the survival curves, the yellow ones, with immunotherapy. Long-term survivors in a subgroup of patients, those having the proper pre-existing immunity. Unfortunately, still, immunotherapy is efficient for many patients, and the era of combination immunotherapy now aims at targeting those tumors and to basically be able to compensate the immune defects with combination immunotherapy. So I hope that nowadays, with immune standardized assays such as Immunoscore and many others, we will be able to do an immune classification of patients. Because today, still, in any given clinical trial that is done based on a specific clinical trial with a specific type of cancer, with a specific mutation on a particular subgroup of patients, the reality of the immune response is that it is very heterogeneous, and that there are apples and oranges in terms of immune response in a given clinical trial. And we better have homogeneous group of patients with standardized immune assays. And with that, I'd like to thank all the people in the lab, very talented postdocs in the lab, all the clinical centers, all our sponsors, the Society for Immunotherapy of Cancer, and to thank all the centers worldwide who validated the Immunoscore assay. And I thank you for listening to this presentation.
Thank you, Dr. Galon, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Now let's get started. Our first question is, do you think your findings on colorectal cancer, CRC, could be applicable to different cancer types?
Yes, indeed, I think. And the success of immunotherapy is actually multiple cancer types. So targeting those T cells in melanoma, in lung, in head and neck, etc., shows that these T cells can be reactivated in multiple cancer types. And also, there are many papers showing the good prognostic value of those T cells to predict the survival of the patient in multiple different cancer types. So having an Immunoscore consensus assay for all those cancer types, I believe, will be very efficient and will be very feasible in the short term.
And what is the difference between Immunoscore and immune signature? Are they redundant?
Yeah, so I mentioned that there are functional immune signatures with different families of immune genes that are associated with the survival of the patient, so stratifying patients into high risk and low risk. So it's a different technology, it's a different measurement in comparison to Immunoscore. Immunoscore takes into account the first three parameters of the immune-contexture. So the type of immune cells, their densities measured by immunohistochemistry, so we are counting the cells, and their location also, taking into account the spatial distribution of the cells. In contrast, the immune signatures is not a cell type density measurement, it's an immune gene profiling. It's not taking into account the location of the cells. So they are complementary signatures, Immunoscore and the immune signature. They are partly redundant because, of course, when patients have a high Immunoscore, this is associated with a better adaptive immunity and a better Th1 response, so they have a better immune signature. But each of the assay has pros and cons, and each of the assay brings additional information. The most relevant and the easiest to be done in routine practice would be the Immunoscore assay.
We have time for one more question. In your opinion, what's the reason why CRC patients are not responding to immune checkpoint therapies?
So that's a very good point. So as I showed you, Immunoscore is not a static measure. Immunoscore and actually the immune response is changing over time. There are more and more immunosuppressive mechanisms. So when the Immunoscore is decreasing and when the adaptive immunity is decreasing, the tumor is progressing, the tumor is invading, which means that at the latest stage of the disease, like patients with metastases, most of them now have a low Immunoscore. Most of them have a lot of immunosuppressive mechanisms in place. So we are remaining at the latest metastatic stage, the stage where the treatment, the immunotherapies are tested and are given. At that stage, unfortunately, most of the patients now have a bad immunity, have a low Immunoscore, have a lot of immunosuppressive mechanisms. So I think immunotherapy will be even more efficient at the earlier stage. And I think that in order to be successful in colon cancer, we need to tackle and to address multiple immunosuppressive mechanisms. Not only one checkpoint will be enough, because not only one checkpoint is overexpressed at the stage 4 metastatic disease in colon cancer.
Well, I'd like to once again thank Dr. Galon for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January of 2019. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.